it's a pleasure to invite you on the stage here, Stephanie Tuminello. I actually apologize for the few English-speaking persons that are here in the hall because we have not translated from Italian. So next time we'll do better. Actually, Stephanie is going to talk about epigenetic clocks, so the second portion of this session. And actually, she's going to talk about a very, very incredible event that happened, I think, in 2001. Yes, the destruction of the Twin Towers. So it's really a very particular context in which she has studied the epigenetics in this study. Okay, here you are.
Hi, mi dispiace perché my Italian is not so good, so thank you in advance for bearing with me while I present in English, and a special thank you to the event organizers for inviting me here today. My name is Stephanie Tuminello. I am a clinical and molecular epidemiologist who works in New York City at Mount Sinai, and today I'm going to be talking about epigenetic clocks and environmental medicine. More specifically, that our environmental exposures do impact our age and our aging process, and that there are different methods, including epigenetics, that we can use to measure this impact. Okay, so as we know from all of the wonderful talks that preceded mine, aging is a complex and nuanced phenomenon. And what I mean by that is that your chronologic age, how old you are from birth until now, does not always reflect your biologic age, the biological aging process that's happening throughout your life course. When your chronological age is less than your biologic age, this is evidence potentially of something called accelerated aging, which means that you are aging, the trajectory of your biologic aging is accelerated, it's faster, it's going more so than what would be predicted by chronologic age alone. And mounting evidence suggests that environmental exposures can lead to this phenomenon of accelerated aging. And this is I'm going to present today some of my own original research that supports this idea. Okay, before we do that, let's just have a quick little recap about epigenetics, what it is and sort of how it works. Epigenetics is the way that our bodies regulate gene expression. So genes can be turned on or off. This is a process that happens during embryogenesis, during development, but can also happen throughout the life course in response to endogenous and exogenous exposures. One of the different epigenetic methods is through something called DNA methylation. You can kind of see here, I've tried to illustrate it. This is sort of a simplified explanation of what goes on, but when you have something called DNA methylation, what happens is you have a methyl group put onto a cytosine at a location throughout the genome known as a CpG site. Cytine followed by the guanine, you put the methyl there. And what sort of happens is if this is in the promoter of a gene, because the methyl group is there, the transcription machinery may not be able to bind, it's sterically hindered, it doesn't fit, in which case the gene will not be expressed. So the gene is silenced or turned off. Because this idea of DNA methylation does change throughout the life course as people age, we can use DNA methylation as a biomarker for aging, and this is the basic principle behind epigenetic clocks. This table that I have here illustrates what we call the first generation of epigenetic clocks, so the first ones that came into existence, though it's worth noting that every day new epigenetic clocks are being made. The first two are the Hannum and the Horvath clocks, and we all had the pleasure of having seen Dr. Steve Horvath present his work yesterday. There's the difference between these two first clocks is that the Hannum method is based on blood. They use blood to determine the DNA methylation signals for the aging, whereas Horvath used tissue. But both correlate more so with your actual chronologic age. It's predicting that. It's trying to predict your age based on your epigenetics, your DNA methylation profile. The later ones, pheno-age, is more so associated with aging-related health span, your health throughout your life. Grim-age is more so focused on mortality. And what you can see at this bottom row is that the different DNA methylation sites that are used, again, these different CPG sites throughout the genome, are different in number and also actually in location for these different epigenetic clocks. So while they're trying to capture similar ideas, the actual DNA methylation profiles that they're using are actually different. Okay, so now to move on to the first project that I wanted to talk about today, which is the World Trade Center event that happened on September 11th in 2001. This is when 2 hijacked planes crashed into the Twin Towers in New York City, and this resulted in, obviously, the towers crashing down. There were fires. It was a large plume of dust and smoke and debris. An estimated 400,000 people were exposed to. Now, not only was this exposure acute, it was also chronic because it took many, many months to clean up all this dust and debris. And so this was what we consider like a very severe exposure event. Now, I'm highlighting here that this dust, right, when the towers came down and all of this dust came about, it contained things that we call carcinogens, so these cancer-causing chemicals, intoxicants like metals, asbestos, PAHs, PCBs, benzene, all of these different things that, again, you don't want to be exposed to, especially because when they were, these things were also epigenetic dysregulators. And what we did was something called an epigenome-wide association study. So because this dust contained all these chemicals, we hypothesized that probably there would be epigenetic dysregulation and also dysregulated aging associated with this WTC exposure. And so we took blood from WTC exposed and WTC unexposed women, and we did, it's called whole epigenome profiling. So we looked at all over 800,000 CPG sites throughout the entire epigenome using this Illumina chip, and we compared how much methylation were at these sites compared to the 2 groups. I sort of just want to highlight here one consideration for this type of work. These were only in females because we were using women from the NYU Women's Health Study to act as controls. Something to consider and keep in mind when you do this type of work is that epigenetics is sex-dependent. Average profiles differ between men and women, and so it's something that you have to take into account when you do these types of projects. And here you can see, because we looked at so many of the different CPG sites throughout the genome, even though the different epigenetic clocks rely on different sites, we had information on all of the sites, so we were able to look at these different epigenetic clocks in parallel. The chronologic age is the first sort of, those first 2 bars here. You can see that the average age at blood donation between WTC exposed and unexposed women was roughly similar, about 53 to 55. This was by design. They were frequency matched on age when we selected our controls in this case control study. And what I want to just sort of highlight here is that no matter which of the different clocks we used here on the x-axis, always we see that WTC exposed biologic age is higher. Especially if you look at the Hahnemann and Horvath clocks, there's even a greater discrepancy, right? It seems that the WTC-exposed women were much older, more so than just a year, by biologic age. And then from there, once we knew their chronologic age and we knew their biologic age for any of these different clocks, we could calculate accelerated aging. Again, that discrepancy in trajectory between how the chronologic age is proceeding and the biologic age is proceeding. You could think of it as a separation in those 2 lines. I want to make a note here that while epigenetic clocks are wonderful, there's an important sort of limitation and capacity that should be noted, which is that the ability of epigenetic clocks to predict age or the aging-related morbidity and mortality is chronologic age dependent. So as people get older, and especially as they reach much more advanced ages, it's more common that it's a phenomenon called DNA methylation saturation will happen, so that these CPG sites will be fully methylated or fully unmethylated, whereas more so in other younger ages, it's usually partially methylated. And when that happens, it's just more difficult and less accurate for the epigenetic clock to pick up the age. And so to account for that, we have to calculate aging acceleration by taking the residual from regressing chronologic age on biologic age, so that we've taken into account these differences in chronologic age at baseline, which is what we did here. And you can see in the model that I've put at the bottom, we also adjusted for things like smoking, BMI, education, white blood cells, batch, to understand how the impact of WTC exposure related to accelerated aging. Another, again, just important caveat, we were using blood, as I mentioned. White blood cells each have their own DNA methylation profiles independently. So again, it's another important consideration for this type of work. And as highlighted here, using the Hannum, Horvath, PhenoAge, and Grimm age clocks, people who were WTC exposed had this phenotype of having more accelerated aging compared to unexposed, though it wasn't statistically significant using the Grimm age clock. Okay, switching gears just a little bit, I want to talk about another project, which is looking at pesticides exposure. So in the US, unfortunately, pesticides are very prominent despite attempted regulation. Organophosphates are a very common type. And what happens is that these will break down into specific metabolites, which can be measured in the urine. And I've highlighted them here. These are the 6 that are just most common. And why I bring this up, both projects in parallel, is because while epigenetic clocks and measuring aging using epigenetics is, I think, wonderful and a super good way to try and do this, there are limitations, as I mentioned, and one of them is that it's also very, very expensive to do so, right? Whereas phenotypic aging, where we measure the biologic aging based on clinical biomarkers, may be better, and they're related to each other. So taking a step back, again, your epigenetics impacts your DNA expression, your gene expression, which will obviously impact your phenotype. So if we look at the biologic aging, if we think about it as a spectrum, yes, you can look at it at the epigenetic part, but you can also look at it more downstream during like the phenotypic stage. And this is what I mean by phenotypic aging. So we could think about these blood-based biomarkers, albumin, creatinine, glucose, C-reactive protein, red blood cell distribution width, alkaline phosphatase, and white blood cell count as other things that we could use biologically to measure biologic aging. And actually, these 9 biomarkers that I have here and that I used in the study that I'm talking about right now, they're the basis for the PhenoAge epigenetic clock. So that epigenetic clock was trained on these biomarkers, and so obviously then there's a correlation between these. And again, the advantage of this is that it's just much more, it's more accessible because of the price. And so we looked at the National Health and Nutrition Examination Study, or NHANES, in the United States. So this is a biannual survey that's given to people in the US, and it's surveyed in such a way to be population representative. And I just want to highlight here, because I think it's worth just illustrating. In the US, we also have an aging population, right? People on average are getting older. But I'm showing here, this is a novel finding, that not only is this aging increasing, accelerated aging is increasing. This discrepancy between chronologic age and biologic age, whereby people are aging more so faster on the biologic level, is increasing over time. And here, again, just to bring this back to environmental exposures, I looked at whether or not having had organophosphate exposure was predictive of this accelerated aging phenotype. And I show here that it was, right? Even after adjusting for these covariates. So people in the NHANES study who donated urine and blood, and for whom we were able to measure the exposure to the pesticides, on average had more accelerated aging than those who did not have pesticides exposure. And the reason I wanted to present these 2 projects in parallel, again, not only because they illustrate this sort of spectrum of looking at biologic aging from epigenetics to phenotype, but also because the WTC project, that blood was collected 20 years out, right? That had been many years past what the exposure event, whereas here with the pesticides, those only remain in the urine for so many days. And so epigenetics and related phenotypic accelerated aging can be used to measure the impact of environmental exposures on aging. It appears sort of on a whole spectrum of time, both immediately after the exposure event and even maybe many, many years after. So these are my main takeaways. Environmental exposures do appear to accelerate aging. Again, they're increasing the biological aging process. They're making it potentially go faster. Epigenetic clocks are useful tools to evaluate this, but phenotypic biomarkers can also be used, possibly in conjunction with these omics methodologies. I think the positive sort of idea is that aging acceleration is dynamic and modifiable, right? And so if we can decrease these environmental exposures, we can hopefully then decrease aging. The caveat to that is if we know people have had these exposures, we may want to screen them for having more sort of aging-related vulnerability at the clinical setting. These are my acknowledgements, and I'd especially like to thank my mentor, Dr. Manuela Teoli, for recommending me to the summit and for also supporting and encouraging this work. Thank you very much.
Very nice, thank you. Are there questions for Stephanie? Yes. Joanna.
Stephanie, very interesting, especially the first one. I think it's a very exciting assessment. So how many years after the incident you have measured the epigenetic clocks? And the second one to the same, how much, I mean, because we also see a big correlation with epigenetic clocks and stress. Could it be also related actually to stress?
Okay, both excellent questions, thank you. Firstly, yeah, it was like 20 years later, right? So this was very much so past the event. Part of that is just because this is, the clinic where I collected the patients from, they're only enrolled into the clinic once they have, it's called a World Trade Center related condition. I think we all know in the US, healthcare is not so good. And so by enrolling into that clinic, they get healthcare and it's sort of the process. So this was after some sort of condition had already manifested. That's why we were able to get them in the clinic and I was able to get their blood. So certainly, yes, that should be taken into account when we think about these results. Your second question about stress, yeah, that's a great point, and I should have mentioned it. Stress itself also causes epigenetic dysregulation. This we know, and we know that people who are exposed to the World Trade Center disaster are more likely to have PTSD, stress-related comorbidity, insomnia, sleeping trouble, etc. It's hard to disentangle those two, to know if it was the toxic chemicals or the actual stressful event itself. My guess is that it's some combination of both, to be honest. I don't think it's even possible to pull them out because that exposure, was just such a complex exposure. But yes, I'm sorry, I don't have a better answer for you. That's just the answer.
Yes, there's a question there.
Stephanie, we have been using GrimAge for now a couple of years, and we have seen, the pack years results are very interesting. It really picks up smokers, okay? But sometimes it turns out positive also in people that don't smoke but live in Milano, which is highly polluted, okay? So the question is, did you measure pack years in this cohort and then split smokers versus non-smokers and see if you had pack years positive in non-smokers?
It's a great question. So firstly, unfortunately, we didn't have degree of smoking. We only knew whether or not they've reported having a smoking history. So that already makes it a little difficult to tease out that association. Here we didn't really see an effect with the smoking, but it's difficult again, sort of to have the same idea. Having said pollution is likely to impact the ability to tease out smoking to epigenetic changes. These people were all exposed to what is sort of a disaster pollution event with smoke and with debris. And also there's a lot of pollution in New York City too. So it's hard. Unfortunately, I don't think here that we can truly get that answer. But I'm thinking of some future projects to try and understand better. Yes, thank you.
Other questions? Actually, Stephanie, I have also a comment for you. Yes, we heard yesterday Steve that said that the epigenetic changes actually can, go back in time. But when perhaps the population is exposed to such a huge disaster, then after 20 years we still see an effect. So this is, we have these 20 years for. I was wondering if it was possible to measure, the quantity of all those chemicals that was present in that time, and sort of make a scale of, diluting them down to have a sort of scale of how much of those pollutants, are correlated then with the epigenetic clock.
Another great question, thank you. Sorry. Okay, yeah, so there's a couple of complicated things there, but I will say, so this work has we've looked at this in mice where we directly expose them to World Trade Center dust, and we see these epigenetic changes like in the immediate and then also sort of more long-term, not as long as 20 years, obviously, but that does sort of support the idea that these are more or less like long-lasting. And we know epigenetic, even though it's definitely true that things are dynamic, epigenetic marks in and of themselves are actually pretty stable, right? In some cases, they're even hereditary, like it can go from one generation to the next. In terms of which chemical exposures, again, another great thing and something that I want to look at. Here we're looking at blood, which makes it difficult. But I think if we could look at this in the tissue, we could also maybe even measure evidence of the different chemical exposures alongside the epigenetic changes and the aging, and that might be able to give us a better answer. Does that answer your question?
Thank you.
Thank you so much.
Okay, so if there's no other questions, I would say that thank you to the speakers for the wonderful presentations. And so this session is closed by now. Okay.